Добрый день, глубоко уважаемые коллеги и гости.
Good afternoon, dear colleagues and guests. Today is the 19th of May 2021. It's 4 o'clock p.m. Moscow time. Not broadcasting. Then let's take the first technical break to settle some technical issues with sound.
Где трансляция идет, там не слышно. То есть не транслируется. В порядке, да? Ну сейчас все равно подождем. Вот, значит, Рогаев здесь у нас только Николай Казимирович. Рогаев тут нижний. Внизу Рогаев, сверху Качан.
Расскажи мне, почему у вас должны быть характеры.
Ну, почему-то он не может присоединиться.
Янковский, он всегда, он на технике.
Понятно? Понятно. А, вот я могу их видеть там. Я этим не управляю. Мне вон, видишь, сколько читать? Я сегодня целый день читаю. Целый день.
Поэтому воды нужно много.
Отвинтим еще, отвинтим. Я говорю больше, чем диссертант. И все вместе делали. Ужас. Я слушала вчера вечером, весь вечер. Сидели, слушала защиту, которую вел Марков, знаешь? Саша Маркова. Физиология. Вот он вел защиту на физиологическом совете по заболеваниям, нейрозаболеваниям, которые на модельном объекте зебрахиш.
И чего?
Вот я целый вечер вчера слушала заседание, ну, чтобы знать, что там говорить, что не говорить и как. Так что учила слова весь вечер.
Давайте скажем, что до кого-то по физиологии. Мы ходим в садик и учим вопросы задать. Мы долго учили, два доктора научили всех. Антиортостатическая поза.
Боже. Ну как там? Все еще. Команды нет, отмашки нет? 5 минут сказали. Ну еще 5 минут подождем. Уже 5 минут прошло. Не появляется Владимирович.
Может, он позвонит на мобильный?
Так она должна связываться, Лариса Викторовна. Это ее миссия. Сейчас я вас буду всех спрашивать.
О чем? О машине.
Представлять и буду, да, каждого представлять буду сейчас. Ну что? Ну что? Где она? Почему она не дает отмашки? Больше 5 минут.
Будем считать, что продолжим, да?
Народу вообще много. На физиологическом совете, по-моему, не было вообще никого. Ну что? Отмашки. Сказали 5 минут подождать, но уже 5 минут, уже 10 прошло.
У нас Янковский не получается со звуком.
Он просит, чтобы его подождали.
Сергей Георгиевич, приветствую вас категорически. Диссертация. Да. Победа разума над костной материей. Так. Николай Александрович, а тут у меня мои коллеги, и они все тоже являются участниками этого диссертационного совета.
Так.
Да.
Дорогие товарищи.
Так, меня слышно?
Коллеги и гости.
Ой, похоже,
вас...
как ни странно, получилось.
Николай Казимирович, вас слышно. Так, итак, технически...
Well, the technical break number 1 is over, and let us continue our session of the Dissertation Council for Defense of Thesis by Nizhnikov Anton Alexandrovich for the degree of Doctor of Biology, Academic Specialty 030207, Genetics, features of formation and action of conformational protein templates in the proteomic prokaryotes and eukaryotes. The order of Academic Secretary of St. Petersburg University, Gniotov Alexander Valentinovich, dated the 11th of February 2021, number indicated. I, Mamon Ludmila Andreevna, Doctor of Biology, Senior Researcher, Professor of the Department of Genetics and Biotechnology of St. Petersburg University, was appointed Chairman of this Dissertation Council. Other members of the Dissertation Council were appointed by the same order, and let me introduce them to you. Pavlov Yuri Ivanovich, Doctor of Biology, Senior Researcher, Department of Genetics and Biotechnology of St. Petersburg University. Savatieva Popova Yelena Vladimirovna, Doctor of Biology, Senior Researcher, Head of the Neurogenetics Laboratory, Pavlov Institute of Physiology of the Russian Academy of Sciences. In the remote access mode, we have Kolchanov Nikolai Alexandrovich, Doctor of Biology, Professor, Academician of Russian Academy of Sciences, Scientific Director of the Federal Research Center, Institute of Cytology and Genetics of Siberian Branch of Russian Academy of Sciences. Nikolai Alexandrovich, can you hear? Can you see us? Yes, I can see you perfectly well. Thank you. Yankovsky Nikolai Kazimirovich, Doctor of Biology, Professor, Academician of Russian Academy of Sciences, Senior Researcher of Genome Analysis Laboratory of Institute of General Genetics of Russian Academy of Sciences. Nikolai Kazimirovich, can you see? Can you hear us? Yes, I can see and hear you with well and with pleasure. And Rogayev Evgeny Ivanovich, Doctor of Biology, Professor, School of Medicine, University of Massachusetts at Amherst, United States of America. Eugenie Ivanovich, can you see and hear us? Yes, I can see and hear you. Right now, I am in Russia. Thank you. The degree applicant, Nizhnikov Anton Alexandrovich, is also present, and the academic advisor of the applicant, Inge Vishtomov Sergei Georgievich, Doctor of Biology, academician, a Russian Academy professor of the Department of Genetics and Biotechnology of St. Petersburg University. To improve the quality of communication, dear colleagues, working in remote access mode, please switch off your microphones, but please remember to turn them on when you are given the floor. Let me inform you that our session is being recorded and broadcast online at St. Petersburg State University website. The speeches are being simultaneously translated from Russian into English. During the live broadcast of the session of the Dissertation Council, an email is currently displayed, to which, during this session, all the listeners may send their opinions and send questions to the applicant online. Questions should be related strictly to the thesis and the ongoing scientific discussion. These questions shall be forwarded to me by our technical support department, and I shall read them during the discussion. Questions should be related strictly to the content of the thesis, and it's necessary to include full name, position, and place of employment of the author of the question. Questions that are not related to the discussion shall not be presented. In accordance with the order of awarding, at St. Petersburg University, degree of Candidate of Sciences and Doctor of Sciences, approved by the local regulations of the university, hereinafter referred to as the order, a session of the Dissertation Council shall be considered competent if at least 2/3 of the appointed members are present, but not less than 4 persons. Our Dissertation Council consists of 6 members, and all 6 are present, including 3 council members in the remote interactive mode. Audio-visual contact has been established with all the council members. Thus, we have the quorum. Let me set forth the following procedure of today's session of the Council. The total duration of approximately 2 hours. First, summary information about the applicant and documents submitted to the Council, and possible questions related to the documents. That will be followed by a summary report of the applicant, where he will outline the key points of his study, approximately 20 minutes. Questions to the applicant strictly on his report, no more than 2 minutes for each question. Answers of the applicant, approximately 10 minutes for all the questions. Speeches of all the council members in turn with their assessment of their study, and the presentation of the applicant with their questions and suggestions to the author of the thesis. Speech of the council chairman with her assessment of the thesis, answers of the degree applicant to questions and comments of the council members. I suggest the applicant to answer each reviewer individually, open discussion and of the attendees, and anyone present may come up with his suggestions to the applicant strictly on the theme of the thesis. All the speakers are kindly requested to register in the registration sheet and introduce themselves fully before speaking. Presentation of questions to the applicant received during the discussion of his presentation, while this session is broadcast online at St. Petersburg University website. Written questions, the presentation of which shall take more than 3 minutes, shall not be presented. Answers of the applicant, speech of the academic advisor, no more than 3 minutes, discussion of the council members before voting, if such discussion is needed. Open individual voting and counting the votes by the Dissertation Council chairman. The results should be recorded in the minutes of the meeting. Make a decision on awarding or not awarding, and closing remarks of the applicant no more than 2 minutes. Well, dear colleagues, do you have questions or objections to this procedure? If there are no questions, let me start the procedure. Please switch off your mobile phones. Yes, I have a question. Opponents, how long will we have? I may tell you. And how much time will I have? Should be brief, and in your review you should focus only on the key points. Okay, so what's my how long will I have? No more than 5 minutes. What's my order? That's my order number. When will I speak? Hold on a second. It's a good question. You will be the first speaker. I see, thank you. So, well, please switch off your mobile phones. And may I ask, what is my number? Yankovsky. You'll be the last one to speak, just before me. Thank you. Thank you. So, well, thank you for your understanding. I answered all the questions. Now let us start with the applicant's presentation. The applicant shall inform us the genetics on the theme features of formation and action of conformational protein templates in the prokaryotes and eukaryotes, was accepted for defense by the order of Academic Secretary of St. Petersburg University on the 4th of February 2021, 88 order number 882/1. So, well, some information about the applicant. Anton Alexandrovich wrote his thesis at St. Petersburg University. His academic advisor is Doctor of Biology, academician of Russian Academy of Sciences, professor of the Department of Genetics and Biotechnology of St. Petersburg State University, Sergei Georgievich Inkevich-Tomov. The number of applicants' publication which set forth the key results according to the enclosed list is 20, in journals indexed in scientometric databases, Web of Science and Scopus. The degree applicant submitted to the academic secretary a full set of documents for acceptance of his thesis for consideration and defense. All the documents comply with Article 12 of Section 3 of the Order. All the documents submitted by the applicant, according to the information I received from the curator, comply with the requirements and are kept in the applicant's attestation file. Copies are available from the officer of the Dissertation Council Activity Support Department, who is currently present. Before I give the floor to the applicant, I have a question to the Dissertation Council members. Is it necessary, dear Council members, do you have any general questions to the applicant? And is it necessary to present and discuss the entire list of documents submitted by the applicant? I see there are no questions, so let me give the floor to Anton Alexandrovich. You have approximately 20 minutes for your presentation. Thank you for your attention. Dear Chairman, dear Council members, dear colleagues, I'm honored to present the paper, my doctoral thesis, the name of which Lyudmila Andreevna already presented. First of all, I'd like to thank all the attendees for finding the opportunity in these hard times to attend, and for two colleagues working in the remote access mode. And let me start my presentation with the central dogma, molecular biology. This is familiar. This triangle got the form in 1970, was formulated by Francis Crick back in 1950, is based on a template principle of distinguished Russian biologist Koltsov. So the central element of this dogma, when applied to classical template transcription translation, states linear reproduction of elements of macromolecule blocks. In this case, the templates are sequences, but discovery of prions in mammals in 1982 by Stanley Prusiner and his colleagues, and then yeast prions maybe by Reed Wigner, published in 1994, complemented this central dogma of molecular biology with another type of template processes, which could be not only with copying elements of macromolecules, but with reproducing information. So templates are information templates, so which Sergei Georgievich calls the matrix processes of type 2. So what happens? The central element for type 2 templates, which this fibrils with special calls beta, is a unique spatial structure, which is a continuous beta sheet of fibril. Here we see transmission microscopy, and what reflects this structure in the right bottom corner, you see the picture is two-dimensional X-ray refraction, which gave the name to cross beta structures. It occurs because of regularly repeated elements in protein. The beta chains, beta sheets spaced at equal intervals produce this picture, which is the golden standards of amyloid nature of proteins. Amyloids, among the most stable biogenic particles, they can spend decades in external environment and preserve their structure, and they are resistant to protease and other acids. Here you may see some other unique properties of amyloid fibrils. Fibrils is good covering them with Congo red. So the golden standard of diagnostics. And of course, an important property of amyloid protein is the resistance to ionic detergents, which makes them different from the majority of known protein complexes. The term amyloid itself comes from starch-like. This historical inaccuracy used to describe starch-like inclusions, and Rudolf Virchow applied it to describe pathological amyloid inclusions in human organs with iodine like starch, and Virchow believed that it's starchy in nature, but after some years their protein nature was demonstrated. The interest to amyloids is explained historically. It's connected with their connection to pathogenetics. More than 40 human proteins have been identified that can form incurable, so-called amyloidosis, and of pathogenic deposits in different tissues and organs, up to full repletion of tissues. Neurodegenerative diseases such as amyloid aggregation, including such socially significant diseases such as Alzheimer's disease, which occurs in every 1 out of 3 patients aged over 85. And before 2000, the history of amyloids is very long, and diseases connected with aggregation of proteins. But back then people didn't know that protein aggregates known, first described in the 17th century. Up to year 2000, only pathogenic amyloids were known, but in year 2000 some proteins were analyzed which are critical, which perform critical biological functions. If we look at the number of pathogenic amyloids and functional amyloids, or amyloid functions, so today we will see that the number of pathogenic and functional amyloids is almost equal, or maybe the number of functional amyloids is greater. What's important, amyloid proteins today are identified not only in humans and animals, but a great number of prokaryotes. In 2021, 33 amyloids have been identified. In 2015, only 8. This shows how quick the progress is in this sphere. If we look at amyloids prokaryotes, almost all of them have been identified in pathogenic bacteria, pathogen host, mostly connected with protein films, coatings. Symbiotic bacteria have not been studied before our study. If we talk about eukaryotes, the functional amyloids, quite well described in mammals, demonstrated biogenesis of melanin and hormone storage. Some stages of long-term memory, for example, are connected with formation of functional amyloids, which are necessary to perform those critical life functions. The white spot on the amyloid map were plants where functional amyloids have not been described, and some groups of amyloids are prions, which infectious properties. Not all prions have amyloid properties, but this is a subject for a separate talk, and in my speech we shall talk about prions connected with aggregation of protein. The first as a human prion, scrappy, which causes a number of neurodegenerative diseases and a number of neurodegenerative diseases in sheep, cows, and other animals. The greatest number of prions, infectious amyloid, connected with aggregation of protein, have been identified in Pekorsky. So we at least 7 prions and yeast prions function as hereditary elements that determine functions, usually considered as factors of protein inactivation. It's believed that prionization leads to reduction of functional activity of proteins in aggregates. And the purpose of the aim of the present study was to identify new features of formation action of protein conformational templates. The research was aimed at solving the following tasks: identify potential amylogenic proteins in plant proteomes, characterize complex of potential amyloidogenic proteins, identify amyloids in proteomes of symbiotic and pathogenic protobacteria species, compare the functional consequence of protein prionization, the genome-wide expression, with the effects of respective structural gene inactivation, study effects of primary structure and production. The following provisions are presented for defense. For the first time, functional amyloid protein was identified in plants, which is a spare protein of seed P. For first time, identified amyloids in symbiotic bacteria, outer membrane of we found the effects of protein prionization are not identical to deletion of this structure. And using the example of yeast transcriptional regulators, shown that the proteins called conditional prions, which acquire and maintain the prion properties only at production levels that differ from the natural ones. And our study, we started with search for functional amyloids in plants. Before we started our work, several proteins have been identified. Mostly they perform protective functions, and also some proteins capable of amyloid, for example, in yeast. But functional amyloids have not been identified. At first stage, a large-scale analysis was conducted, which included almost 3 million proteins from 76 plant species, and we found that with QPIN1 domain most enriched amylogenic regions, and QPIN1 domain is typical for spare seed proteins, which is one of the important components of our so then the detergent resistant in pea seeds, which we selected for our hypothesis, and we found this detergent resistant proteins which form stable aggregates, spare proteins, and so we identified this domains. This beta barrel structure, and consists, as you can see, of several beta sheets. In vitro, we demonstrated that Isolane and its domains form typical fibrils, typical for amyloids, when dyed in Congo red of the green light, which proves its amyloid nature. Then we proceeded to in vivo testing, and on cross sections of cotyledons and cotyledons. So we identified those protein aggregates because Isolane is accumulated in such membrane particles, protein bodies, and found that Isolane there in the form of detergent-resistant aggregates, which manifests its amyloid nature. We dyed these protein bodies in Congo red by refrigerants, and we selected conditions where it was possible to perform microscopy. For that, we had to remove membranes from those protein bodies and spread them with ultrasound a little bit to see their inside beta fibril structures which bind the antibodies was marked with colloid gold. So this proved amyloid nature in vivo. Then we studied what happens in dynamics of amyloid isolane. So we found that they are accumulated as seeds. So there is a system in seeds which disassembles them. Unfortunately, it doesn't exist in our brain, and unfortunately these amyloids are very stable, resistant to proteolytic peptine and pancreatine. They are preserved in pea seeds, and prospective strategy is new species with reduced content of protein, these protein aggregates. This isolane in this condition has expressed toxicity both for animals, humans, and for mammal and human cells. Fibril component is highly toxic, and non-fibril practically is non-toxic. Then we shifted our focus to the role of amyloidogenesis on interorganismal interactions, and we decided to study symbiotic bacteria because symbiotic bacteria, amyloid protein, were not found in amyloid proteins in them. So we studied the rhizobia, was important for agriculture, that can improve nitrogen nutrition. And at first, so we conducted analysis of rhizobia and identified associations of regions, and in experimental protein screening rhizobia bacteria, we identified 54 proteins with highest count, ROPA, with a transmembrane protein with structure predicted beta barrel. So we used in our analysis ROPA B protein. We found that in vitro, those both proteins produce amylofibrils with birefringence. It was interesting to see what happens in vivo. In vivo, those both proteins present mostly in aggregates, occur in aggregates, detergent resistant. They're resistant to high temperature, which shows their great stability. Typical for amyloids, which is very typical and interesting when stimulation flavonoid imitates initial stage of symbiotic interactions. The number of aggregates ROPA increased and size increased, which manifests the possibility of amyloid in formation of symbiotic relations. We managed in vivo to demonstrate that the capsule of these bacteria, rhizobia, dyed with amyloid-specific dyes, demonstrate by refringence. We demonstrated immune electronic microscope and formation of fibrils on the surface, to which the antibodies against these proteins bind. So we described two amyloid proteins for the first time. Then we studied pathogenic bacteria. I will not describe in details since we are short of time. So we also identified a number of proteins with beta barrel domains in symbiotic pathogenic bacteria, amyloidogenic. So such beta barrel domains in amyloidogenesis. So we tested the properties of one of proteins and for its domain, and so we indicated in vitro in detail its amyloid properties, and we demonstrated that if its domain is superproduced of E. coli, fibrils are formed on the surface that also show amyloid functions. A work was published which demonstrates amyloid properties of protein in vivo. So that's a new amyloid, a new group connected with peptidase functions. And here, of course, we get this question of functionality, produce amyloids. And when studying this issue, we used yeast prions because yeast prion is quite well studied, and in yeast you may conduct various experiments. So we worked with 3+ prion, the component of global regulator of transcription, then evolution regulator, which affects expression of most yeast genome, and we demonstrated that this prion has manifestations similar to annihilation but different by some features. So we compared transcriptomes, yeast transcriptomes, because it changes during prionization. And first we found that it has greater influence on transcriptome than prions. We also saw that there is topology, and so the first chromosome is superexpressed against repressed, which is connected with coded components of ribosome. So then we compared prionization effects by biological processes that they affect, and we found that it has specific manifestations of full repression of components of in expression of all translational factors, while prionization doesn't affect it. And the paths that are changed with prionization, activation of some metabolic paths, such as which does not occur in deletion. So, and since I cannot talk about the other experiments, but generally speaking, we were able to demonstrate considerable differences between prionization and deletion. It's not identical deletion. So, and finally, the last section, which I very briefly want to describe, is a conditional prions. Here we studied GLN3, the transcriptional of nitrogen, which has OML-genic, contains prions, is overproduction. It forms aggregates. If we overproduce full-length proteins, no aggregates are formed. But if we co-overproduce this domain with QPN plus, and in small it performs aggregates. So we decided to explore these aggregates, the loss of plasmide for overproduction doesn't result of full GN3, and aggregates are stable in mitosis, but they are eliminated when overproduction stops. So this aggregated state is inherited but supported only occurs only during overproduction. So effective elimination of GLN3 as well as which controls most yeast prions. Moreover, we managed to register the difference of GLN in prion strain, which we described, where it formed typical amyloid detergent resistant aggregates, while in strain where we saw no aggregates, so we also described some manifestations for this prion, which such as increase in resistance of strain, and we demonstrated it's infectious, which is one of the key properties of prions, yeast prions, this GLN3 with protein transformation. So yet its unique property is that meeting the yeast, it only with overproduction of structural protein, which is novelty of this prion, and we suggested such prions, to call such prions as GLN3 conditional, because they exist in such unnatural conditions, and other prion proteins, call them genuine, and the third group are artificial prions with modified primary structure. The conclusions are shown in my thesis in this slide, so let me not read them, but let me briefly say, finish by saying that in this work, for the first time, we describe functional amyloid proteins in plants, amyloid in symbiotic bacteria, shown that domains of beta barrel structure are in amyloidogenic, and in prionization protein is not identical, and a new group of conformational protein conditional prions was described. Thank you very much for your attention. Thank you, Anton Alexandrovich. Now we will proceed to an important stage of today's session, to questions. Who has questions to the applicant? Please, Alexei Petrovich Galkin, please introduce yourself. Please say your name, position, rank, etc. Thank you. Good afternoon, dear colleagues. Alex, Deputy Director of St. Petersburg Branch of General Genetics Institute, Professor of Department of Genetics and Biotechnology of St. Petersburg University, Anton Alexandrovich. I have a short question for you. You described amyloids, bacterial amyloids, and you E. coli amyloids. Now experts are using CDAG system, which is based on CSGA protein bacteria, which can be prionized, that can be replaced with any other protein, and explore its amyloid properties in intercell space. But this system implied that no other amyloids can be observed, otherwise this system won't work properly. So it seems that your question makes this belief invalid. I pressed some button here. Can you hear me? Yes. Yeah. E. coli, we studied the domain. So we did not analyze the properties of full-length protein. We all use this CDAG system, and this data is shown in CDAG system of production of domain corresponding to signal to the surface of a cell. The work which I mentioned, where colleagues studied properties of full-length protein, which is now has a different name. It shows formation of what they say functional fibrils in deep stationary form of biofilms of E. coli. Fibrils, they have not performed a diffraction analysis, but they performed a version of analysis, and they claim that these fibrils have beta sheets, so they are similar to amyloids, but they found this only in one strain, in deep stationary state, at the stage of forming biosheets. So for strains that we use, I think the CDAG system can be used in strains where it works. We are working with other proteins, and we know very well that the majority of strains, even laboratory strains, have bright red coloring, which means they produce quite a large number of other amyloid fibrils. Thank you. As I understand, you are satisfied. Yes. Yes, I'm fully satisfied with the answer. Thank you. Do we have other questions? Lyudmila Alexeyevna, welcome. Please introduce yourself. Thank you for your presentation. Here is my question. Formation of amyloids in seeds. Maybe it's good for the seeds. Proteins are preserved, but maybe it's bad considering the nutritional value of peas and maybe in other cultures. Thank you, Lyudmila Alexeyevna, for this question. Let me give you answer in two parts. The domain QPIN1, these are the key amylogenic in peas domains. They are very rich in amylogenic regions, so that's the specific feature, and most enriched spare proteins in lotus. And then I read an article that seeds can wait 1,000 years, but judging by when pancreatine so their concentration is even higher, it's pure protein, amyloids survive. That means that yes, they significantly reduce nutritional value. So maybe for plants this conservation mechanism is good, but for us this is a negative feature which modern science can eliminate. Thank you. Are you satisfied? Yes, I'm satisfied. So maybe this indeed is one of the signals for features can be used in selection. Professor of Department of Genetics and Biology, head of laboratory of genetic engineering of plants. Do we have other questions? Then let me ask a question which I have not included in my review, but preparing for this session I thought about the genes that have this triad normal loss of function. So what do you think? Is this typical for all genes, or there are some proteins with some functions where this phenomenon will prevail? We found something similar because in our laboratory we for the functionality of this protein, spare protein, and lectin, which defends against pathogens. There is data, Isolane, which is spare protein in seeds, which is stored for some reason in huge amounts, is produced in rhizobia. So maybe in root nodules. So maybe this is the answer to the question of polyfunctional proteins. But I'd like to say that in development genetics, this term is widely used, mostly genes that take part in inter-cell interactions. So think about this. Maybe the existence of such effect for proteins involved in interaction. Thank you, Lyudmila Andreevna. Who else has questions? Kolchanov, please tell me. Stress. amyloidogenesis, are they connected? Is there any connection? Maybe from your research between stress and amyloidogenesis, reduction of amyloids. In plants, at least, amyloid proteins in vivo, formed in vivo, in normal conditions, which we consider normal, we have never found. So maybe my answer is that there is no such connection in plants. But if we look at other organisms, maybe formation of amyloid aggregates is a typical formation of protein aggregates is one of the effects observed in cells in case of stress. The disturbance of folding systems, so then they produce amyloid aggregates are accumulated, and the cell systems should disassemble them. If they manage to do that, if they fail to do so, then various pathologies occur. Many diseases connected with amyloidosis occur, mostly in case of mutation of regulatory parts of genes, production of proteins involved in development of this amyloidosis. So that's a very typical situation. Thank you. I'm fully satisfied with the answer. Thank you. Nikolai Alexandrovich. Thank you. Please introduce yourself and ask your question. Yankovsky Nikolai Kazimirovich, Head of General Genetics Institute of Russian Academy of Sciences. I read the thesis which made me ask new questions. What you said about high resistance of Isolane to various influences that may think the function is to keep this seed inedible, but the seed itself has capacity at certain phases to change the conformational properties. So what the seed can do itself, maybe this is not a way to find out how they transition from this very stable protein to unstable condition accepted for the plant itself and could be used for technological purposes. So maybe our brain will become younger. Thank you, Nikolai Kazimirovich, for this very interesting question, which indeed is connected with the medical part. Of course, certainly. So we are starting this project, which is quite big and complex, because deciphering such cell systems is quite hard, especially in plants, which have huge genomes and proteomes. And what I would like to mention is we observe dramatic reduction in terminating seeds, nanomeres, the proteolytic regions. That means that aggregates are disassembled, or maybe that's chaperone. So we identified one chaperone, CPC, the chaperone, which is similar to yeast chaperone, yeast prions, which is interesting. So maybe it's him, but there is still a lot of work to do. in this sphere. And if we are successful, if we identify such factors, we may think about the theoretical possibility of using them in some biomedical models, and then later. Thank you, Anton Alexandrovich. Nikolai Kazimirovich, are you satisfied with the answer? Yes. So when we meet, I will ask you many questions. Thank you for your answer. I'm satisfied. Thank you. Please ask your question. Please introduce yourself first. Glinin Timofey Sergeyevich, junior researcher of genetics department, candidate of biological sciences. It's a very interesting presentation from the point of wide views of amyloids in different kingdoms, but there was an interesting comment about the possibility to use the functional significance of amyloids in seeds to make them preserve their ability to germinate longer. But theoretically, what could be the significance for bacteria using these amyloids? You demonstrated that in rhizobia they are also present. So how can you explain this phenomenon? So maybe you obtained some data or you have some ideas about this. Thank you for your question. Well, first, let me say that there was continuation of this work, which I did not include in this thesis, in candidate's thesis, Anastasia Kasabapova, who will present her thesis shortly. And in our lab, we found that these amyloids are formed in bacteroids, the terminal form present in nodules, and contains lots of fibril proteins. And considering these effects in this flavonoid, which imitates the initial state of symbiosis, we thought that they are active at the initial stages. Here we see the increase in the number of these aggregates, but maybe they are needed for mechanical stabilization of cells in nodules. Maybe, for example, for some signal interactions with a host organism. So here there are many possibilities. We are just starting to explore this, but already now we may say that in nodules where differentiated cells of these bacteria are present, large quantity of these proteins is observed. Thank you. I'm satisfied with the answer. Thank you. Who else has questions? Siganov Viktor Evgenievich, head of laboratory of molecular biology. You said that amyloid forms of proteins are present, but differentiated bacteroids are typical only for part of that form nodules. And do you know anything about not differentiated in forming nodules, in non-defined nodules? Thank you, Viktor Evgenievich. The only thing we have done, we used as mutant. So this is an artificial system, because it's not quite clear what happens in reality. But morphological differentiation of bacteroids does not occur. The cells look similar to free living culture, and so there large number of these fibrils is also produced, and birefringence is observed. So these are technical details. Thank you. I'm satisfied. Do we have more questions? Have we received any questions online at this point? No, we have not. Then let's move to the next stage of our work, presentation of external reviews. Let me not read the full, but only focus on the key points, in my opinion. Of course, all the reviews have been published at St. Petersburg University website. If you're interested, Doctor of Biology, Professor of Institute of General Genetics, Zakharov is the positive review. In his review it says that several decades yeast remained the main model for study of prions. Considerable let me finish reading. As yeasters of Department of Genetics of St. Petersburg University, under the guidance of Professor and Anton Alexandrovich started his study of yeast and then widened the scope and the conclusion, which is also present in all the reviews. I will read it now and will not read it again. The thesis by Nizhnikov Anton Alexandrovich corresponds to the main requirements set by the order. Order number is given, and the degree applicant Nizhnikov Anton Alexandrovich deserves being awarded the degree of Doctor of Biological Sciences, Specialty 030207 Genetics. Articles 9 and 11 of the above-mentioned order has not been violated by the applicant. Then, all is well, please continue. Then, we will not take a technical break, but we will continue with the reviews. The next review was sent by of Federal Research Center of Fundamental Basis of Biotechnology of Russian Academy of Sciences, Professor, academician Popov Vladimir Olegovich, Doctor of Chemical Sciences. The review is possible, has no critical remark, and the thesis is well written, is a complete study conducted with modern method that has started a new chapter in the study of prions, amyloidogenesis in general. The next review was sent by a senior researcher of the molecular genetics of Bach Institute of Biochemistry of Federal Research Center of Biotechnology of Russian Academy of Sciences, Doctor of Biology, Kushnirov Vitaly Vladimirovich. The review is positive and it complements the achievements of the applicant, and the work describes a new class of yeast prions, conditional prions, that manifest prion only at overproduction. Hence, one comment: it was earlier demonstrated that protein also can form amyloids with overproduction. Salnikov, year 2005. But these amyloids have not been considered classified as prions because their maintenance depends on other prion pin plus. So these amyloids have not been inherited, but very quickly created on the basis of pin plus. So it would be desirable to test and eliminate such a possibility for GLN3 prion, but this has not been done in, and Salnikova work has not been mentioned. The work is well illustrated with experimental results, but some drawings, the title is found on another page, and this could be easily avoided. The work is classified by a wide range of approaches, and the author has used methods of bioinformatic, transpectology, and microscopy. The work with yeast, the applicant changed to plants and bacteria. This means he has capacity and skills to necessary for a manner of research processes. And finally, one last review sent by candidate of senior researcher, professor of School of Biological Sciences of Georgia Institute of Technology, Atlanta, Georgia, head of laboratory of amyloids of St. Petersburg University, active member of American Association for Promotion of Science, Yuri Olegovich Chernov. The review is positive, and the author provides detailed analysis of the thesis by chapter, and says that chapter 4 is dedicated to analysis of protein aggregation GLN3, which the author calls a conditional prion, since it aggregates only with overproduction. So one of the main criteria of prion nature of GLN3 loss of aggregation when chaperone is activated. But the strain contains another prion, and this prion maybe determines aggregation of GLN3. Aggregation of polyglutamine, zone 1, in yeast cells, which depends on energenic prions, this is spin plus. The zone 1, though capable of aggregation, reproduced prions in yeast cells is not. And in my opinion, the data do not prove, though allow to suggest GLN meet the criteria of independent prion. In this context, the term conditional prion is questionable, and infection is not of yeast prions. Prions are defined as transmissive proteins. That is why it's sufficient transmission in cell generations, but without overproduction. So LSB2 aggregation, of which is inherited without overproduction and doesn't depend on PIN plus, meets prion criteria, unlike GLN3. It should be said that these comments only apply to the term, and the data about GLN3 aggregation are interesting and are of certain. While reading the paper, I got a number of questions to some details. First, how does the author explain pseudo-negative results when testing protein cubin 1,2 in system CDAD? Second, the drawing figure is in fact in this experiment did not produce yeast spheroplasts, so it's not clear how Isolane aggregates penetrated, and if they penetrate inside the cell, how in this case the toxicity of amyloids can be explained. Third, if yeast YGHG has amyloid properties in vivo outside CDAX system where it merges with artificial anchor. It's absolutely not, it's not clear, while reduction of gene expression of translation mechanism and ribosome RNA should result in nonsense suppression. What is the suggested mechanism? And 5. It will be interesting to compare the author's conclusions with the later published work of Lehman Lee laboratory that also studied prion SVI1, an expression of genome yeast. These comments and questions apply to works and do not reduce its scientific significance, and by no means question the main conclusions of the author, supported by a great number of experimental results. The author published 20 articles in highly rated journals, and the list of publications would provide good support in American University Research 1 category, which in my opinion is the American equivalent to Russian doctoral degree. Thank you for your attention. Dan Alexandrovich, will you please answer questions and comments of the reviewers? First of all, I thank Yuri Olegovich and Vitaly Vladimirovich for reading my work and sending their comments. So let me start with questions by Vitaly Vladimirovich Kushnirov about inherited amyloid, glutamine 3 never produced in overproduction. So we screened and such clones were not found. So PIN3 does not induce aggregation, but in presence of PIN3, PIN plus, which induces induction of full-length protein. That's the first thing. And second, in this work, we didn't give this information, but we analyzed the status PIN of protein transformants produced, transformation of GLN3 minus and GLN3 plus strains, and as far as I remember, in 3 out of 7 transformants with GLN3 status, RNQ protein did not aggregate, which means that they are PIN minus. So as we expected, aggregated state does not depend on GLN3. Salnikova I should have quoted, of course, but then on the other hand, honestly, I wouldn't say that these phenomena are similar, but they have different mechanisms. And as Vitaly Vladimirovich noted, such amyloids, they are not prions. And here we are dealing with protein. Now, Livina Andreevna, do I have to, or can I take both reviews at the same time? As for Yuri Olegovich, he has a similar question about a comment quantity of the production with PIN plus. I think this is the same work of Salnikova, published in 2005. The story is similar here. The answer would be the same and let me say again that PIN plus acts as an inductor, and aggregation is not inherited, while in GLN3 aggregation is inherited. It occurs regardless of PIN plus, because we can support GLN3 in strains PIN minus, and PIN plus does not induce aggregation. Yuri Olegovich also has a question about amyloid properties of YGHJ, and a study was published by another team that it forms amyloid-like the question about pseudo-negative result in CDAC system. I especially included this slide, QPN1 domain, which has amyloid properties with Congo red media. I think this is connected in CDAC system for some reason. It does not form non-linear fibrils, but some aggregates. Maybe they demonstrate such birefringence. They are different from typical amyloid fibrils, maybe because of toxicity, because QPN1, 2 is highly toxic, especially in amyloid state. Maybe this occurs. And the question about toxicity, how can we explain toxicity of Isolane? I said that it has sugar binding domains. With eukaryotic cells, they destroy the membrane and bound with polysaccharides on the surface of fungi and bacteria, and their mechanisms are not quite clear, have not been properly studied. And there are two more questions. Let me answer them very briefly. Why reduction of gene expression should lead to nonsense suppression, which is reflection of lower reduction, because in all molecular systems such imbalance results to some reduction of function. So in translation, aminoglycoside antibiotics make it inactive, which is similar to expression reduction, and nonsense suppression occurs, and efficiency of translation is decreased. And our results in transcriptomics compared with Lyman Lee work was published recently with different strains in different conditions, and this key effect that we described is they also achieved full inhibition of translation apparatus, but the gene spectrum was very different, only 30% similar, so which pleiotropic effect manifests the pleiotropic effect, which regulates. So hopefully I answered all the questions. Thank you very much. We got a suggestion to take another technical break, maybe for 5 minutes or maybe for 10 minutes. So how long shall we? Let's break for 10 minutes. Let's take a technical break for 10 minutes.
Дорогие коллеги, мы...
Dear colleagues, let's finish the second technical break and proceed to the next part of our session. Speeches of the Dissertation Council members and their reviews. I suggest the following order we should start with Kolchanov Nikolai Alexandrovich, Rogayev Evgeny Ivanovich, Savateva Popova Elena Vladimirovna, and Yankovsky Nikolai Kazimirovich. He'll be the last reviewer, and the last review shall be presented by me. Dear Nikolai Alexandrovich, you'll be the first speaker. Dear colleagues, the thesis by Anton Alexandrovich is dedicated to the study of formation, organization, and functioning of amylogenic proteins in plants and microorganisms. First of all, about relevance of the work. Despite the active study of the process, there is a number of open questions, such as factors of the environment activating formation of amyloids, the functional significance of amyloid genesis, its nature, and understanding if amyloid genesis is a failure of cell program, or is this a programmed cascade of events which have an important significance for the organism, and specificity of amyloid genesis. And in relation with this, the relevance of the thesis is beyond any doubt as compared with other groups of plants. One of the key has not been studied in the sphere of biology of amyloidogenesis. Among the most interesting results of the thesis, which have high degree of novelty and significance, in my opinion, is the following. On the basis of analysis and experiments, a functional amyloid protein has been identified for the first time, which is a spare protein in seed pea seeds. So this spare protein is accumulated in the form of amyloid aggregates, which are toxic for insects and fungi. And amyloidogenesis of spare proteins potentially can secure seed protection from pathogens. For the first time, Rho A and Rho B proteins are true amyloids involved in formation of symbiont host systems. So this is a systematic approach of a higher level. For the first time, as prion, transcription regulator of yeast, which is hereditary in several generations of cells. So this is to answer the question, what are the conditions of amyloid formation? The results of analysis of some plants verified by experimental data led the author to the conclusion that functional amyloidogenesis is a conservative characteristic not only of beans but of a number of plants. And the overall conclusion is significant, demonstrates that prion conversion as conformational template of does not lead to full protein inactivation, but results in the change of function of structured gene, but can be seen as a form of mutation of gene. So what I would like to emphasize from the point of methodology, to answer the questions, the author, who has lots of experiments with experimental work, uses comprehensive approach, which includes and this increases the depth of the study conducted in the work, because the results of the analysis described in the thesis enable to choose the ways of experimental studies. And the practical significance has already been mentioned, and first of all, it has a great significance in this sense because the results obtained by the author, spare is an important component of human nutrition. So what I particularly liked, the thesis is compact, but the presentation is very logical. The author has clearly communicates his ideas. The language of the thesis is good. The title fully reflects conducted studies. Results make a significant contribution to the study of diversity of amyloids. The author first identified in plants and symbionts, and validity is beyond any doubt. It has been supported by 20 publications in 15 leading and 5 in professional magazine biochemistry, so the thesis fully reflects its content. And in conclusion, the thesis by Anton A. Nizhnikov in eukaryotes corresponds to the requirements set by the order on awarding academic degrees at St. Petersburg University, and the degree applicant Anton Alexandrovich Nizhnikov deserves being awarded the degree of Doctor of Biology, specialty 030207, genetics. Thank you. Thank you, Nikolai Alexandrovich. Since there were no critical remarks and questions, I suggest we proceed to the next review by Yuri Ivanovich Pavlov. So, good opportunity to remove the mask. I will allow myself to focus only on the points which have not other speakers have not touched upon yet. Previous reviewers mentioned that Anton uses many different methods, and what should be mentioned, it would be impossible without his organizer talent, who found a group of young talented researchers who can conduct these studies, established contacts to researchers other researchers in Russia and abroad, and that is very important. And no wonder that the study was supported by 4 independent structures, for example, in the US. So this is something that would be the first thing mentioned. I was impressed by the number of international conferences. 50 conferences without publications. That's very interesting. That's impressive. That's very impressive. The introduction impressed me because quotes start with 19th century publication. This demonstrates the overall scope. So then the theory of Maury's, I have a question about Maury's theory. The distribution of prions is evidence of this theory and not vice versa, as the author claims. What is important in the introduction, everything is all is well, but there is one problem. The first picture in the described structure of amyloids So, but without any illustrations, and without an illustration, it's possible to understand that, but it's quite hard. But now, at today's session, everything was perfect. Well, I have some doubt that the statement submitted for defense is well formulated. For example, provision submitted for defense identification of prion. I don't think this is something, but if prion is functional, so that could be submitted for defense. So I would phrase it differently. About the first chapter, we learned a lot, and I spoke to Anton, and I found why he mentions allergy. So I have no critical remarks here. As for the lethal effect in chapter 2, we have also discussed. Now I am satisfied and I know what is meant. Now, in chapter 3, as it seemed to me, it's written for professional geneticist of yeast, but to somebody with different background, it would be difficult to understand this chapter. Next, in chapter 4, well, I liked these experiments very much. We found that there are analogs to his experiments, and but Anton explained quite well how his experiments are different. So maybe these impressive experiments were so exciting that the author for a moment lost his sense of style, and they were tasted gabloid status. And so this is one of the strange phrases, because otherwise the thesis is very well written. And in conclusion, let me say that a careful reviewer would say that all these results have a descriptive nature. The mechanisms of these phenomena have not been discovered, and for an age grant that would be a big disadvantage, because it's considered it's necessary to study mechanisms. I don't think this is a drawback. I think that will enable to start. Of course, the author discovered many new phenomena, and this work has great potential, but I won't read the formal part, which has been read before. I have a very high opinion of this work. Thank you, Yuri Ivanovich. Anton Alexandrovich, will you answer? Yes, thank you, Yuri Ivanovich, for your detailed review. I agree with all your remarks, and I will certainly take them into account in my further work. I did not understand. Please let me clarify. There are no questions. So the questions, I asked my questions in the review, for example, about the mechanisms. If you could talk, there are three hypotheses. How will you test? Because these, you just made this hypothesis. It's not clear how to make a choice between the hypotheses. Thank you for your question. The question is connected with Rob A and Rob B proteins and how they can participate, and we have actually discussed that already. I have three hypotheses in my thesis. The initial stages of symbiosis, maybe binding with skeletal proteins or interacting with plants, the signal interaction with plants. As for the initial stages of so the data we obtained, it contradicts that these proteins are typical for initial stages of symbiosis, and so maybe they perform they have the stabilization function already in bacteria nodules. So maybe this hypothesis is the signal role of amyloids is well known for RIB3 and RIB1, programming necroptosis of functional amyloids and prion this. But in this case it's unlikely because we see very stable, rigid structures which do not change. And as for the experimental tests, here there's some difficulty, so we are thinking about it. It's one of our projects. If you look, so almost all the structures of these proteins, the sequence of these proteins is in beta sheets, so they don't have specific amylogenic regions, which makes it hard. Here we have the entire gene delete, and so we are thinking about that to describe the primary function, and then we will probably do more targeted work, more focused work. This will be my answer. Thank you. Thank you. If I answered all the questions? Yuri Ivanovich, are you satisfied? Yes, I am. That's good. Then let us proceed to the next review by Eugene Ivanovich Rogayev. Dear colleagues, can you hear me well? Yes. The thesis by Anton A. Nizhnikov is dedicated to the study of various aspects of conformational templates, fibrils, and their activities. I liked the title from the start because it does not say amyloid and prion word, and it doesn't say about the biological functions but actions, because the biological functions often the term effect and action often replaced with the incorrect term of biological functions. So here everything is very well described. The thesis itself is very well written. I'm not familiar with amyloids outside of mammals, so it was very interesting for me to read this work, and everything was quite clear. The work itself is conceptually complete study, published in many articles and international journals from the first quarter is necessary. The work is published in from my own experience with this magazine, this journal. One of the rules of this magazine is over 3 reviewers, sometimes more than 5. So I am sure all the conclusions set forth in this part of the work have been tested very well. So these publications prove validity. As for provisions and goals are quite well set. Maybe provision number 3, effect of prionization, is not equal to, could be, the wording could be changed, as the prionization effect is not equal to the loss of gene function, because here the authors talking about the process, and gene deletion on yeast. But in this case, of course, the greatest interest is the work to study amyloids in plants, the discovery of from informatics to the study, experimental study of amyloid nature of plants. This work is a pioneer as amyloid properties in seeds. So the author has identified made interesting observations of spare proteins. So that's quite interesting. So proteins, as we understand, protease resistant, are very stable, and what molecular mechanisms contribute to this such elimination, it would be very interesting to find this out. So maybe the author already has some information about this, or is already thinking about this. For example, how the involvement of chaperone of disassembly of spare proteins in seeds, and the activation of what protease occurs, something on a molecular level. So such work asks to be performed. And in the course of discussion, some questions have already answered. So I will miss them. For example, how the author explains absence of full-length glycerin in protein positives of germinated seeds. What could be seen in Figure 7. Of course, there's a question about pathogenic nature of such structures, properties of such natures, how they exist in other plants, how they act, the toxicity of such fibrils in mammals or to bacteria. Was toxicity tested of bacteria structure? So in this case, the author's talking here. We are talking about functionality. It's interesting that according to the author, in nodule in rhizobia form amyloid symbiotic partners in beans. This is interesting from the point of functional role and the work itself. The thesis have has opened a new path. So I think this thesis certainly belongs to this category, and so something can be research could continue with on the basis of this the results of this work. So I will probably drop some points connected with questions the applicant has already answered. Maybe one thing should be mentioned: the study of prions of yeast. When the author writes about that yeast transcriptome is different to some degree, I think this is quite obvious if we don't go into details. So, for example, what are the molecular mechanisms, loss of function, amyloid structures, maybe not only amyloid structures, but monomer forms of protein. Maybe they partly have this function in amyloids signal pathways of loss of function, of gene function. So maybe here makes sense to give more details, and so I will not talk about terminology and the works that experimental models that have already been discussed, such as CDAC and yeast as a conditional prion, how different it is from prion-like in this context. Maybe the only question here, my only question will be how or to what degree how often for large number of proteins expressed in large how often prion properties will be observed in yeast proteins GLN3. In general, I may say that the thesis by Nizhnikov, Anton Alexandrovich, on the theme features of formation and action of conformational protein templates corresponds to the requirements set by the order on awarding academic degrees at St. Petersburg University. And the applicant deserves being awarded the degree of Doctor of Biological Sciences. Thank you. Thank you. Eugenie Ivanovich. Anton Alexandrovich, will you please answer? Thank you, Eugenie Ivanovich, for such detailed analysis of my work. I agree with all your comments, of course, about the questions that you asked. Let me start with toxicity. Yes, across biology, we conduct an experiment on toxicity on mammal cells, carcinoma, I think, and if I'm not mistaken, and I can actually show you the slide. The toxicity was quite strong in all the versions in glycerine, this glycerine, and two isolated domains. All three showed great toxicity in mammal cell cultures, and they're aggregated in their starting points where they produce aggregates but not amyloids. So toxicity is specific to as for bacteria, now we are conducting such experiments, and toxicity seems to be universal to fungi, bacterial cells and mammal cells, and is connected most probably, if we talk about mechanisms, of sugar binding domains, lectin, the membranes or eukaryotic cells, which creates toxic effects in fungi and bacteria have toxicity, but the mechanisms are not well known. Now, as for while many proteins have in various experiments, bacteria, maybe because this is a more important amyloidogenic determinant feature. The molecule mostly consists of beta sheets, as one colleague said. So maybe you don't need big changes for it to transfer to amyloid state. As for how common these proteins are in plants, cupin is probably one of the oldest protein domains present in humans and in plants, with spare and protective functions. And in the bean family, I don't want to give you invalid information, but they all barrel-like domains, and they are many. As for mechanisms specific to prions, well, in reality, we should not see prions as regulatory elements, and now there are many works I have not mentioned where prionization is not connected with aggregation of protein. They demonstrate various versions of modification, and the biocondensates are many works demonstrated by condensate, and so they some evolutionary adapted molecular systems emerge where prions act specifically, and if such bifunctionality when monomers. So this is a question still. So maybe such cases may also occur as a kind of a switch, because what states in SPIP protein work, but with SPIT amyloid properties have not been demonstrated, and prion properties, though it resembles a switch. So maybe that's all I have to say. Let me clarify your answer. About toxicity, you said the aggregates and non-amyloid, and then you said fibrillogenic, and then you said amyloids. So look, is there any information what degree of fibrillogenity polymer so what is non-amyloid aggregation? Thank you for the question. The matter is that these substances in water solutions, because they have hydrophobic beta barrels in water solutions, they start forming aggregates which are not structured, not amyloid. They don't have amyloid properties. So these unstructured aggregates at the initial point are non-toxic. Then, under certain conditions, you can obtain structured amyloids, and these structured amyloid fibrils are toxic for mammal. So here we show some yeast test. They are highly toxic, as lethal, some of them. So that the fibril state of amyloids is toxic. They're not fibril. They are. And finally, what I would like to add here, if ultrasound destroy fibrils, break fibrils into small particles, when size becomes smaller, toxicity also goes down. So these fibrils behave as in a similar way. Hopefully I answered your question, Eugene Ivanovich. Yes, I'm satisfied. Thank you. Thank you. Okay, Eugene Ivanovich. Thank you, Anton Alexandrovich. Then let us proceed to the next review by Elena Vladimirovna Savateva-Popova. Dear Eugene Ivanovich, for me it's an honor to speak after you. What was said by other reviewers is it is quite unique. This indeed is a new chapter, and in order to write this chapter on a great number of experiments, as was mentioned, that were mentioned already, here you need great talent, and I think everybody can understand that, everybody can see that. Whoever this talent was obvious during our preparation for the Congress dedicated to 100th Jubilee of our department, and what Anton Alexandrovich did was very impressive, and Engevich Tomov. And thank you. Let me thank all the speakers. I will drop a lot, but an excellent provision. So provisions 3 and 4 spiralization of the structure and deletion of structural protein, everything mentioned here. And then the next about conditional prions, a long time ago, mutants, because under one conditions it's absolutely normal, and under different it's absolutely an absolute full. It's full under the normal temperature. Normal temperature is impossible. It doesn't 100% has inclusions, and only stress HSP 104 plays a significant role here. It's not only just a factor. No, of course, this I shouldn't talk about scientific novelty. Practical significance I like very much, especially where seeds can be a source of food allergy. Myself, as somebody who's allergic to peanuts, I was very touched by this practical consideration. Validity is obvious. How many works Squirtle 1, sometimes up to 5. And of course, everything is valid. And then, for some reason, it was not mentioned that the author won a number of awards from the government of St. Petersburg, and recently, together with Anton A, won an award from the President of Russian Federation for young scientists. So these are recent events. I think this shows highest level of reviewers, unlike us, all 6 of us. I beg your pardon. I'm allergic not only to peanuts. You know, it is true. For the first time, after so many years of reviewing different works, I have no critical remarks. I maybe following the tradition and the genre of it's a detective of Anton Alexandrovich's thesis is a detective story. Everything, the conclusions. I want to admire this detective story, this thriller on prions. Wonderfully written. In the beginning, he says that amyloids could be one of the most ancient structural and functional organization of biological micromolecules at the start, when life started on our planet, and this gradual demonstration of how it demonstrates the evolution is an important agent that performs a very important functions necessary. So it was impossible, of course, to the 75 species here analyzed to animals found, and Eric Kandel was the first to mention it, who described the cytoplasmic protein, that prions are necessary to change in the synaptic plasticity and to preserve long-term memory. And this happened a few years ago. Ingevich Tomov, I gave me some imprints. So now, and that's really so. Very powerful. That's a very powerful function, and the way it is, demonstrates the following. So all organisms, and think of hereditary Vavilov law. You can obtain anything. Could be yeast, could be plants, could be E. coli, and various animals. And one of the good illustrations is a work by Rogaev. Let him correct me. I came across in 1995. He identified from a small plant, Arabidopsis. It's tiny. At your department, at our selection department, so such a plant, so the beginning of Alzheimer's disease. So the plant cannot demonstrate the loss of spatial orientation. So we started with this, and then it happened that, well, I would say that is an excellent, the work is very well written and the results very well presented. So, indeed, I don't have any critical remarks or questions. So this happens because when reading the work, any question is immediately answered by the author in the next paragraph, and I have never come across this before. So this is really impressive. And so maybe there is only one question for historical, political reasons, but could be explained by the author. So here is your work is an absolute of homological of Nikolai Vavilov, but you said that is the result of analysis of 75 plant species, spare proteins enriched with amyloid regions. So amyloid. A lot was said here already. And then there is a question. If it's really so maybe so what's the role? So the question, if it's really so, maybe we are not getting close at a new level, to the new level of knowledge, to Lysenko's, according to Wikipedia, because we don't have other sources. It's hard for us to. But initially, this Wikipedia says that Nikolai Vavilov, but then rejected by Michurin. So it's a reaction to of seeds to stress and HSP 104. So this is a question which does not reduce the thesis by Nizhnikov as a wonderful piece on the the theme fully corresponds to the requirements set by the order. Shall I give the date? At St. Petersburg University, and the degree applicant certainly Nizhnikov Anton Alexandrovich deserves being awarded the degree of Doctor of Biology, specialty 030207 genetics. Articles 9 and 11 of the above-mentioned order has not been violated by the author. So I think that this is an outstanding, excellently written, excellently summarized as a great gift to all of us, whatever you call it. Thank you, Elena Vladimirovna, for your review. Anton Alexandrovich, Will you please answer a single comment made by thank you, Ludmila Andreevna. So this was probably a question. Thank you, Elena Vladimirovna, for such a detailed analysis of my work. Well, of course, we have not started as was what we, though of course this study, many researchers are doing this around the world, unfortunately, but in reality there are other works recently published. Such transcription regulator was identified that geneticist in her work demonstrated this acts as prion in yeast, as transcription regulator. And several months ago, another work was published, vernalization, transcription regulator L3 Arabidopsis has prion, and at low temperatures it suppresses growth, and when is the temperature conformational in this prion domain, and it starts to aggregate, and it works as transcription repressed that ensure growth and blossoming. So this is quite similar to this phenomenon. According to the latest data, such regulation exists. One thing I want to say: these are not prions. This is a prion-like phenomena, but it's not prions. Otherwise, thank you for your review and your question. Thank you. Thank you. Let's move on to the next review by Nikolai Kazimirovich Yankovsky. Nikolai Kazimirovich, will you please read your review? Can you hear me? Dear colleagues, do you mind me not wearing a face mask? Thank you. Well, this is a very rare, a very unique case that a work is so good. It gives you lots of pleasure as you read it and as you listen to this discussion, and you get questions, and so questions related human genome. I've been working 30 years and reading this work which links all these things together. You start thinking that maybe I thought about, and this work activated many long-gone spheres of interest. I will not read the formal part of the review, and as already presented by Dr. Kolchanov and repeated by other reviewers, I wrote some questions which I asked in writing, so let me read them to hear the answers, but mostly what was already said by other reviewers I won't repeat. But here I'd like to give some consideration, because I saw this and I don't know if these will be questions, so maybe that will be interesting. As for an interesting phenomena of all those protein aggregates, they do not exist without a genome and transcriptome, afternoon and proteome, but polymorphism, and without genome and proteome, it does not genetic polymorphism. It exists on proteome level, and from it's impossible. But the environment determines and genetic and in this respect, this brings us to makes us remember Lysenko, academician Lysenko, because the nucleotide sequence does not change, and but allele changes by the environment. Never mind. So in this, so because of that, it's interesting. So the first chapter is about amyloid proteins in plants, and here I agree with what that I saw. So according to is the world priority, the author described for the first time. But looking at this part after the presentation, I have two questions. Let me ask another question already asked by Dr. Rogayev. Eugene Ivanovich asked, do they have this spare proteins? There are many, so it could be, but so, or do all the paralogs. But no, here I found that amylogenic is different, and I also agree that by the style accuracy, genetic accuracy, which is quite rare, the work is excellent, and I really rejoiced at it. So as for paralogs, so if they have connected with amylogenic properties or not, and so the questions in the text for which I would like the author to answer is one of the questions is property of other. But there's another question: the allergic properties, are they connected with formation of amyloid aggregates in seeds or not? So there's the first question. The second part, and it's very impressive that the applicant covered all the passed through all the stages of experimental on objects, and most people never work on such diversity of objects such as plants, then bacteria, E. coli is very specific, and then talking about amyloids and their role in other and in humans. So this characterizes the author in various fields of genetics, which is especially valuable because each study and synthesis in each work is especially valuable, especially today. Speaking of rhizobia, it says that the first time such structures, such quarter structures in the alternative state, but let me share one thing with you. Maybe in 1991, bacteria, when I studied bacteria, I heard Jimo Shapiro at Chicago University. He conducted a simple experiment. He studied how different genes of E. coli work because the dish dries, and how the colony, the cells are colored, but one gene is affected instead of its own transcribed, and how obviously it's blue. Is it all blue? The colony in the stationary phase has the multi-cell organism with own structured tissues, allocation of colored sheets, and the depth of colony for each gene is its own in symmetry. I found this extremely interesting, and I don't know, maybe everybody knows this already. I have not read about this for about 8 years. And on these rhizobia, so maybe it's the same genes work. So, as bacteria, of course, will be repressed, and in this respect, it never occurred to me that I knew that colony will be multicolored and structured, will have internal structure, but I never related this, that will be an analog of a multi-cell organism with differentiated tissues, which will be have colored differently inside a colony. And listening to this and reading this thesis, I jumped 30 years back, so it would be interesting how this how this was used. And so I have a question which I have to ask to get an answer. So the structures, the spatial beta barrels, and there's a question. These beta barrels, it's not clear if they are the stage as bacteria were not different from bacteria, or later, or maybe earlier. So if that's true, then in humans maybe these beta barrels could be identified, as the author found them in rhizobia and in humans. And in the final section of the thesis, the author who said infectious amyloids and significance is quite, because it's not mentioned, but first thing is, I was impressed that spider web and silk, these are amyloids, and which means that the structure and regulator is a regulator, maybe in the phase of growth. If it works as a regulator, but when it reaches the stationary phase, it becomes a structure, protein, allele state in development, in a stationary phase development of a colony, something. So maybe I would say something. So since I'm an academician, I can do that. And here, this prionization of proteins can be a regulatory mechanism, a functional activity at the protein. The idea is very interesting, and maybe it is true, but it seems that here the number of various manifestations of this gene is rather great, and it could be as a regulator, but then maybe it also functions as a regulated object when it turns into amyloids. At least in the case of peas, is a different structure, different mechanical properties, and maybe from the point of this, for those who's going to eat it. So maybe it's just a method of the type of antibiotics, or maybe for some specific opponents. This is a, I don't eat peas, but I'm not an insect, so maybe. And once again, that the discussion is what that could mean. It makes a great impression, the depth and the quality of applicants' training. Of course, not so they leave no doubt that the thesis, all the answers are here, but such a thesis as now and defense gives you a real pleasure. and I would like to congratulate the council and various, and I see that this form, the form of today's defense, is interesting because, at least for me as a participant, and I think maybe something new will be said by the opponents, something interesting for the subject itself and for Anton Alexandrovich. And conclusion is that I will not read the final section. I hope we all have the same. And so let me congratulate the applicant in advance. Thank you, Nikolai Kazimirovich. Anton Alexandrovich, can you answer? Yes. Thank you, Nikolai Kazimirovich, for reading my thesis and for your interesting ideas and suggestions that I need to think over. Let me briefly answer the questions in your review. So first of all, if we plan to study other spare proteins, legumines of pea seeds, they all have domains similar to QPIN1. So this domain they are quite different in primary structure, but the spatial structure is similar, and they are typical for beans. And so we are working with other classes of spare proteins, and the law for Isolane just seems to have amyloid properties to locate together Isolane, but ligumine seems to be not as so well structured. In vivo, I think in amyloid properties it won't have. Even in vitro, it behaves as a poorly structured protein. How it's connected with allergic properties? We discussed already that there are three components, obviously. Let me start that Isolane is the main peanuts, as Isolane of peas, the main allergy gene in cases of being allergic to peanuts. And Isolane has sugar binding activity, so we may suggest that lectin activity is binding with, though it's not clear to me, but lectin can bind to cells, eukaryotic cells, and such bonds generate allergies. And second thing is protease resistant regions or food allergies. Such three component system we still need to study and understand further. And as for connection with beta barrel proteins, we have already discussed. Let me say one thing, that humans also have beta barrel domain proteins connected with mutase associated with sclerosis. They also have beta barrel and amyloid peptide beta, considered to be one of the main conditions of Alzheimer's disease. I should be careful here. In vitro oligomer, and they have high toxicity because as they build into the membrane and violate, break through membranes. And prionization phenomenon, which you mentioned in your, I don't know how well it could be, how wide as amyloid prions, there are not that many, but let me say that in recent years many works have been published connected with prions, could bind not only with biocondensates, amyloid in nature, and honestly I have not realized them as maybe they are prions, maybe. And these biocondensates, they seem to be quite common, and they, in yeast at least, the common regulatory mechanism. So the potential of prion regulation is quite high. So the problem is that we know little about it. Hopefully I answered all your questions in the review. Nikolai Kazimirovich, are you satisfied? Nikolai Kazimirovich, are you satisfied? Please turn the microphone. Yeah, can you hear me now? Yes. Yes, I'm quite satisfied and thank the applicant for the pleasure. Let me thank you and the colleagues. I think it was a very interesting discussion. It was very interesting for me and hopefully for the others. Thank you very much. And the last review is mine, and since I have the privilege of being the last reviewer, I will not give an overview of the work. I'll just say a couple of works that we had a chance to learn about. Proteins have similar spatial structures, which has a number of common features, and the approach which I liked very much. So the work is bioinformatic work which enabled the author among great number of proteins to identify those that later were studied with wet methods to determine their properties and to understand if they belong to the amyloid class. And what I would like to say that the achievements made by the author are impressive. The list of publications is really impressive, which outlines the main results obtained by the author. And in general, I don't have any critical remarks in a review. The formal phrases are there, and at that I will finish my speech. I have one question. Is anyone present willing to speak about is there anyone present here willing to speak? Yes, please. Dear colleagues, let me not take your attention. Anton Alexandrovich for many years has studied, wrote his candidate's thesis under my guidance, and today it was a pleasure for me to hear his presentation, especially considering what's happening in our sphere now. Since the beginning of this century, we found that prions, our amyloids, is not something that kills our brain, our kidney, but there are functional amyloids, and the interest to this theme has increased. Lots of junk, lots of pseudo sensations. Today CBAB protein was mentioned. This was one of the main mystifications. It's not a prion, it's not an amyloid. So why am I saying this? Anton Alexandrovich states everything very clearly. What are amyloids, what are prions? He always remembers how they are different, and he answers correctly to all questions. And there are many sensations. As for prions and regulatory function, classical prions is easy how to understand how to include them in any regulator, turn on, turn off, but it's not clear how to turn them off. And there are many junk papers on this, but this work is not junk. It makes a significant fundamental contribution to the study, because first of all the first amyloid, functional amyloid of a whole kingdom of plants. And we can also, I can mention other details, but under every conclusion of this work I would put my signature. This is honestly a very good work without any pseudo sensations. Thank you, Alexei Petrovich. And Igor Anatolyevich, please introduce yourself. Please turn the microphone on. I beg your pardon. Tikhanovich Igor Anatolyevich, Doctor of Biological Sciences of Research Institute and the Dean of Biological Faculty. I think we could congratulate ourselves and first of all Anton Alexandrovich on his excellent work, and let me say a few words how this work was performed. It's important to understand today's reality, and the reality is that it's necessary there's a need for integration between science and education. Anton Alexandrovich demonstrates how it should be, the way it should be. Indeed, he's an excellent scientist, he's a talented teacher, and in a sphere which today is one of the most in demand, bioinformatics, he teaches, he provides all the programs on bioinformatics at our faculty. I'm lucky. So to achieve something in science, it's not enough so you also need good luck, and such good luck, I think, is in that Anton Alexandrovich became a member of the well-known school led by academician Ingevich Tomov, where many excellent Mikhail Teravanisyanov, now late, and Anton managed to get the necessary skills to work on a high fundamental level. But then he came to the Institute of Agricultural Microbiology, and this work, of course, the work of the institute was connected with doing something useful for agriculture, and Anton came to the laboratory where the idea of interaction was dominant, and still it remains dominant. So it's very logical that how microorganisms, and he was able to demonstrate that before they met, plants, microbes don't know about each other, and the first signal that microorganisms get, these are flavonoids from plants. Lutein was mentioned already, and when rhizobia get this first signal, they respond by synthesis of certain proteins, which Anton and Asprion like amyloids. So why is this? Is it necessary? Why is it needed in the process of interaction? Of course, I cannot answer this question, but let me emphasize further: for microbes to penetrate plants, it's necessary to create a channel, and such proteins, as it seems to me, so maybe if maybe this the structural component of such structures needed for bacteria to move inside plants, and thus this work is an integration between St. Petersburg University and the educational has an educational component, and agricultural in institutes of Russian Academy, and Anton combines it all and provided a new quality, new of research, new type of research, which received very high marks, very high rankings from the reviewers, which will certainly make St. Petersburg University famous and Institute of Agricultural Microbiology famous. So let me thank you and wish you lots of success in your future work. Do we have more questions? Is anyone else willing to speak? Galina Anatolievna, please introduce yourself. Zhuralova Galina Anatolievna, professor, the Department of Genetics and Biotechnology. Can you hear me? I just wanted to say other speakers did not mention. First of all, Anton maybe is one of the youngest doctors who defended his thesis at our university, at least in biology. Because Anton, how old are you? 34, 33. I'm 33. Well, so this age, if we look at Moscow University, there it has quite a number of young doctors. In St. Petersburg University, this is something that's very rare. It's people in their 50s. It's hard to persuade them. For Anton, it was very important. It's an excellent example to the young. I remember when Anton first came to our department, and we were honestly afraid because he was so active, and everybody, all our employees said, no, this is too much. And as a result, he came to Peterhof, and then everything worked perfectly well. Another talent was mentioned that he communicates well with different people. So many of these people I know in person, but it was impossible how he managed to bring together people from so many different cities, from Kazan, from Novosibirsk. This is another important talent which is useful to the young researchers, which he teaches. So the art of getting grants. He won so many grants. The ratio is very high, maybe not 100%, but still high. That's because he writes well, and secondly, he gives very good names. So there was something about cells. So such a name in itself already is a key to success. RNF by title. So that's also very interesting, because there are titles that draw attention, and usually Anton's titles play a very major role. And as I have one wish, before I finish, Anton, I always admire how much you can accomplish. You have the number of tasks of your duties is growing, and you are chairman of the Scientific Commission, editor-in-chief of Biological Communication Journal, secretary. So you should save energy. Please stop, because I'm really worried about you. And let me wish you further success. Thank you, Galina Anatolievna. Is anyone else willing to speak? Maybe online we received any questions? No. Then let's give the floor to the academic advisor of Anton Alexandrovich, Sergei Georgievich Ingevich Tomov. Dear friends, there are three things you can. It's all hard to fill it. Talking to a friend who came from a long journey, eating, and scratching where it itches. And so I cannot, as an academic advisor, I can only talk about the applicant himself. All these years working with Anton Alexandrovich was a pleasure and was highly productive, and I was always impressed by how he managed to combine in-depth interest and his civil position. Let me give you some examples. Academic secretary of Vavilov Society for many years, including, and it was mentioned today, he prepared the International Congress in Genetics dedicated to 100th anniversary of our genetics department. Since in the beginning of this year, he became editor-in-chief of Biological Reports journal, other serious biology, St. University newsletter. He's the head of laboratory at the Institute of Agricultural Microbiology, and so maybe these are I mentioned only some things, and he manages to combine research with educational activities, because this is really where he works with doctoral students and graduate students. So this characterizes the applicant. It makes one wonder how he manages to find so much time. He has more than 24 hours in a day. And in conclusion, I'd like to thank Anton for pleasure of working together. I hope I will have this opportunity in the future and wish him success in the future. Thank you, Sergei Georgievich. Once again, let me ask if anyone, any of the guests are willing to speak. So before we vote, let me ask all the members of the Dissertation Council if the Dissertation Councils or the degree applicant any critical remarks or any comments regarding the procedure connected with working in the remote access mode. Then let us proceed to the next point, is voting. And let me ask the council members, is it necessary for us to discuss the results? And the sound shall be switched off. No, we don't. Oh, that's good. Then, if we don't need such a break, let me ask everybody if you can hear if you can hear me, and if you can hear us, if you can see us, because we will proceed to voting shortly, and voting shall be open and personal. We need to vote if the Dissertation Council need to decide on awarding on the degree to Anton Alexandrovich Nizhnikov, the degree. Then, if no one has any objections, then let me ask each council member individually for his opinion about the thesis. And Anton Alexandrovich Nizhnikov, if he deserves being awarded the degree of Doctor of Biology. Kolchanov Nikolai Alexandrovich, your opinion. I'm for awarding the degree to the Doctor of Biological Sciences. Pavlov Yuri Ivanovich, your opinion. I have the same opinion as Dr. Kolchanov. Thank you. Rogayev Eugenie Ivanovich deserves. Yelena Vladimirovna Savateva Popova. Yes, certainly the degree should be awarded. Yankovsky Nikolai Kazimirovich, your opinion, please? Can you hear me now? Yes. Yes, of course, the degree should be awarded, and I remember in a National Dissertation Commission, and I remember there was opportunity to make. So I think this is, it is possible. The thesis should be, in my opinion, of course, coincides with the opinion of other members of the council. Thus, dear colleagues and friends, let me inform you that out of 6 voting members of the council, 6 voted for, no one voted against, and nobody abstains. The decision to award to Nizhnikov Anton Alexandrovich the degree of Doctor of Biological Sciences, Specialty 030207 Genetics, has been made. And let me give the floor to the applicant for his closing remarks. Dear colleagues, for me it's a great pleasure to thank, first of all, my academic advisor, Dr. Tomov, who at all stages of my work assisted me, and I hope that we will continue doing research together. I am very grateful to Igor Anatolyevich Tikhanovich, who supported this work, and as Igor Anatolyevich said, this work indeed is a synthesis of 2 outstanding schools of research, Department of Genetics, where I started my work, which turned 100 years in 2019, and the Institute of Agricultural Microbiology, and the laboratory, this of which I am the head at the institute, is 130 years old. I am very grateful to our laboratory, to Antonets, to Dr. Belousov, Yuri Viktorovich Malavichka, Marina Igorna Belousova, Anastina Legamna Kasalapova, Nizhnikov, and other colleagues, and thank them, Alexei Petrovich Galkin, who was my academic advisor as a candidate, my master's thesis, which was quite recently. And let me thank my colleagues with whom we've been working together for a long time: Galina Antonovna Zhuravleva, Dr. Bondarev, Elena Alexandrovna Andreeva, Pavel Alexandrovich Zikin, and dear colleagues from the Institute of Cytology of Russian Academy of Sciences, Anna Igorevna Savaska and Maxim Igorovich Savatsky, Oleg Nikolaevich Demidov. Unfortunately, I won't be able to mention everybody. I just want to thank the Institute of Microbiology, because probably there is no single laboratory with whom we have not worked together. I beg your pardon if I forgot to mention somebody. The list of colleagues I would like to thank is really long. And of course, I'd like to thank the grant givers, Russian Scientific and the Ministry of Science and Education, and our wonderful committee and resource centers. And thank all my dear family, my friends, without whom this wouldn't happen, because here, of course, usually if things are not good at home, so then this work I won't be able to do this work. And also I'd like to wish scientific and pedagogical success. So thank you very much for working together all these years. Thank you. Thank you so much. Thank you, Anton Alexandrovich. At that, I declare our session finished, and I thank everybody for the participation, all the council members, all those who took part in discussion. I'd like to thank those who supported the technical support department and our curator, Larissa Viktorovna Khrushcheva, for her invaluable help. And thank you